seminar. So today is a great pleasure to have Pedro here in this seminar. So Pedro is PhD at Técnico, working mostly on 2D materials like perfume and plasmonic, etc. And then took a bit of a turn and is now at the University of Amsterdam working on active matter. So Pedro, thank you very much for coming and for this.
Thank you so much. Thank you for the introduction. It's a pleasure to be here. So yeah, so I took a bit of a turn, but you will see that most of the frameworks, in a sense, of most of the tools are quite similar in the way that overarching questions are very compatible, at least in my mind. But so today I will talk about some recent work that I've been doing in collective dynamics with broken parity, with some chirality induced in the system that we will overall call hot viscous materials. And I will go a bit into detail on that, and I will show you both a bit of rheology and a bit of biology to give you an overall idea of what I've been doing lately. But so the idea behind our research now is to think on how can we design and think of the chiral microscopic properties of a system to induce some desired rheological response, some desired microscopic response on our system. So in a sense, we try to induce the system with the property that we want through the chirality of the system. For that, we will imagine that our fluid is constituted by an ensemble of particles that you can think that rotate or that they broke parity in some way. Mostly today we'll be thinking in somewhat uniform systems, but in the future we also want to expand to more non-uniform systems. And so far there's already been some experimental works in the field. Some of them quite famous by now, like the work of Nick Factory on sea urchins, embryos, where you bring together an ensemble of rotating particles, be it driven or active, and ranging, as you can see, from very wide types of systems, from magnetic to biological systems. And when you bring these systems together, they start to develop interesting macroscopic properties that are inherited from this break of symmetry at the microscopic level. So just to give you an overall idea of what's been doing in the field recently. And so to give you a more detailed idea of what I'll be talking today, so I thought of splitting somehow this talk in three main parts. So I will introduce a bit better what I understand by hot viscosity and by hot rheology, and then I will show you first two examples on the theoretical side of what we can do with these kinds of systems and what are the responses that we foresee for the systems. And then I will talk a bit about our experimental implementations that we've been working on in Amsterdam. But so to make it clearer what I'm talking here about breaking of chirality and what viscosities and what transport coefficients, the idea is that if you have a system where you somewhat break your parity inversion, and this can be done on several ways, but usually you can think of having a system like this cartoon here, where you have internal torques on your constituents, where you have some transverse forces that act on your particles, Or also you can also think that due to the rotation of your particles, the collisions are somewhat parity broken, like in this example here. So if your parameter impact changes sign, your scattering angle is different. So if you have some of these ingredients in the system, then what happens is that you will inherit a different type of symmetry, and that then you can study formally doing a gradient expansion or doing Chapman install on your kinetic theory, and then you can obtain your transport coefficients as usual in a sense, but with the difference that now you are not so constrained by the symmetries. You have new symmetries in the system to play with. And the aspect for today is what happens to viscosity, what happens to your viscoelastic tensor under these conditions. And what happens, especially in 2D, because of course, as I was saying, you need to break your parity, but you should still have a well-defined momentum operator. What I mean is that it shouldn't average to zero. So if you are 2D or axisymmetric, what happens is that you have now not only your usual shear viscosity that couples shear deformations to shear stresses, but now you have this odd viscosity here, eta per, that off diagonal, that couples shear deformation to normal deformations at the linear level. So the key ingredient to think about odd transport coefficients is that they show up in this off diagonal terms, and this is inherited by the rotation that you have at your microscopical level. Then, of course, the specific details of your coefficients will vary with the system, but the overall form is the same. And then, with this new structure of your stress tensor, you of course change also the properties of overall of your fluid, of your Navier-Stokes. And of course you can see it both in compressible and in incompressible cases. Today we'll be focused in incompressible, or at least we are aiming for incompressible scenarios, whereas also the compressible scenarios also have interesting phenomena. But particularly in incompressible, it can be shown that you can rewrite the effect of the viscosity as, in a sense, as a pressure gauge, where you use the vorticity of your system to correct, in a sense, the pressure. Which, of course, immediately means that then boundary effects or boundary conditions are essential for certain flow, and in particular boundary conditions that are not simply velocity boundary conditions. So it will be critical to understand the stress boundary conditions on your system to see the effects of this odd viscosity. And so, with that in mind, what we started to do was to look into the effects of this odd viscosity in capillary waves. So in surface waves with on a fluid with both odd viscosity and surface tension. And in a way, this is also natural, not only because of the necessity of some stress boundary conditions, but also with the overall idea that then you can possibly in the future control the deformation of your system. You can have a droplet and control its shape, or you can have a flow and control the propagation and shape of your surface waves on the system. And also, if you, and we will see it in a bit, both odd viscosity and surface tension induce cubic dispersion on your system, so it's natural that they have a strong interplay at the waves developed. And to look into these systems, I will be using both the natural or shear Ohn-Zorn number and also what I call an odd Ohn-Zorn number that measure the relative ratio between the viscous dissipation and surface tension and odd viscosity and surface tension. And it's also, by the way, important to note that odd viscosity is non-dissipative because it's off diagonal. And so we did this both in theory and also in our simulations. And what we see is that, okay, if you run our simulations without any Ohn-Zorn number on a periodic boundary condition, you, as expected, you just have a standing wave of the surface waves. But if you start to add some odd viscosity to your system, you see three, I would say, three main phenomena, three main characteristics that happens on your system. Well, first and very evident, I would say, you now have some chiral directed propagation. You see that you can control to go either right or left depending on the sign of the odd viscosity that you choose, which ultimately derives from the rotation, from the internal rotation of your constituents. So that is one that we would love to understand. The other aspect, and maybe let me rerun, is that and if you will, how well you can see it, but if you look at the bottom row, the vorticity that develops near the interface, near the surface, is wider in the cases with odd viscosity compared to the one without odd viscosity. Let me run this. So you see, this is very evident. I would say that you have a strong and constant vertical boundary layer on your system, and also, and perhaps more striking, on the last point, you have a weird Stokes drift happening here. That, as you can see, if you place a Lagrangian particle on your system, it is advected, which you can say, okay, but that is kind of expected because now you have a propagating wave. But interestingly, and if you note, for relatively low values of odd viscosity, this odd Stokes drift, if you want, occurs in one direction. But if you crank up your odd viscosity sufficiently high, then there's an inversion, and then you start to have the Stokes drift in the other directions. So these are the three, if I can, odd phenomena that you see in the systems. And so let's try to see how can we make sense of that. Starting by the easiest in a way, by the chiral propagation.
So can I ask you something that maybe I missed that, but what are you simulating? What is the model?
The model is incompressible Navier-Stokes with the stress tensor that I showed you before.
Because it says chiral, how did you add chirality to...
At the level of the odd viscosity.
Okay.
So, back. So I'm just saying that besides the shear component, we have also technically we are doing that in the simulations with this effective pressure and in the bulk, whereas of course at the interface boundary conditions, then it's with real pressure and not the corrected one. But
there is something missing in this problem, I think. Because you
probably.
Okay, in the parentheses you have a vector and then you have a gradient of a vector, which is the tensor. And then
no, yeah, so it should have, yeah.
Then it's projecting, then it's a vector. It's a gradient.
It's a gradient of the vorticity. So yeah, this parentheses should be get.
Okay.
It's the gradient of the magnitude of the vorticity, right? Because the vorticity in 2D is a Taylor scale. Because it's always directed either along or contrary to Z.
Okay. I don't see it's.
Okay.
There is probably some mistake.
There's a, yeah, okay.
I don't know.
Well, yeah, the ball is back there, yeah, you can see that. Yeah, so into the chiral propagation, we can understand more directly if you look at the dispersion relation of these waves, and you see here that this onsorb is what breaks the symmetry of your dispersion relation and starts to deform your left propagating and right propagating branches of your dispersion relation, which is not so evident for small onsorb numbers, but as soon as you start to increase it a bit, it becomes quite clear that you have a quasi-acoustic, quasi-linear branch and quasi-quadratic branch on your system, and then of course the interplay of these two propagation modes then result on you having the propagation in only one direction. And you can also see that on our simulations, if you leave a packet of waves to evolve, as we did here, and you can Fourier transform this propagation and get exactly the dispersion relation that we were hoping for, where also it's important to note that it's not only the deformation on the real part of the dispersion relation, but also the quadratic branch here has also a way higher diffusion, has a way higher decay on the system than the propagated linear mode that is stronger here. And so our theory can reproduce very well our simulations, our simulated results, where you can see here there's no fitting parameter between what we get into our simulations and our linearized dispersion relation here. And so this already explains why you have propagation that it's dominated by one of the modes. But then the fact that one of the modes dominates over the other also explains the increase of the boundary layer on your system. Because you see the vorticity boundary layer in your system will grow also with the frequency of the mode as indicated here. But while in pure surface waves you only have one mode, or two equivalent modes, now we have two unequivalent modes. And so that means that one of the modes leads to the decay, to the confinement of the vorticity boundary layer, while the new one, or the other one if you wish, the one unlocked by the viscosity, leads to the growth of the boundary layer thickness with increased hot viscosity. And this so it explains why you see exactly this growth of vorticity boundary layer. Then, and to the third point, we think that this is quite connected with this strange Stokes drift that we observe in the system. So this is to be not yet fully understood, but what we believe is that it is exactly this increase of the vorticity boundary layer near the surface, then leads to this increased Stokes drift. That, as I was telling you, also the most striking, this is for me the most striking peculiarity, is that for small odd viscosities, so for small Onsor numbers, or odd Onsor numbers, you propagate, you drift in one direction, and then when you increase sufficiently your odd viscosity, then you change the drift. And bear in mind that meanwhile, the wave is propagating always in the same direction. So you just change it if your drift is going along with the wave or contrary to the wave. And then we studied also a bit of statistics on that, or did a more complete study of the case, and you can see that it seems to exist a critical value of Onsor number above which you have positive drift velocities of an ensemble of Lagrangian particles that you track, whereas below you have all of your Lagrangian particles drifting in the other direction. We still have to understand this a bit better, but we really believe that all of this is really due to enhanced nonlinear effects due to the odd viscosity, because you can also see that what I'm telling you is in fact not only dependent on your Onsor number or on your change of Onsor number, but also on the amplitude of your wave, so on the amplitude of the initial conditions that you impose. So there's not only this critical Onsor number, but there's also somewhat critical amplitude above which you really unlock this this anomalous, if you wish, drift or drift change between left and right.
Can I
Yes.
ask you two things? The first one is where you decide where you look at the tracer, because it's slightly below the average position of the wave.
Yes.
Is there a reason for that? And then second, what controls the period of the modulation? So basically, you see that when it's moving, it's basically rotating,
Yeah.
while being dragged by the wave, right? What is controlling the time it takes to do one turn?
We haven't looked into that yet.
Because it looks like being a competition between the velocity, the time it takes for the wave
Yes.
to go a distance that is comparable with the perimeter of
Yeah.
this, the diameter of this thing and the time it takes to rotate. So it's just a competition between the two time scales. I don't see why you need non-linearities to explain that. So if you could explain how long it takes to rotate and what controls that.
Totally fair. But then you would still see it for small amplitude waves.
Except if the waves are not long enough that the shear induced by the fact that you are at the wave, you have a length scale there. It's compared to the I also believe that it's not going to do it all the time, right?
No,
so,
of course, yeah.
So basically, you need to induce some kind of shear to have an effect on that, and this
Yeah.
is related to the amplitude of the wave.
Yes,
Because
of course.
and the length scale that you need to compare with is the diameter of these loops.
Of the loops.
And that's what makes things non-linear, sorry,
Yeah.
because if the growth depends on this other parameter, then the size of the boundary layer depends on the amplitude of the
Okay,
wave.
but
So
yeah.
in that sense, it's non-linear.
Yeah, okay, but you can explain it just by comparing time scales. The difficulty
yes, but-
is then to say all these time scales depend
Of
on
course,
the OH,
yeah.
but
Yeah.
I don't need to make it so complicated.
I see.
Yeah.
That's my guess,
I-
but
Yeah, I agree.
But it's totally fair. I agree as well. Yeah. Okay, but so this is what we've been doing on capillary waves, and so as you notice here, so far I'm letting my shear and my odd viscosity being just a constant parameter of the system. But now we wanted to also to look on how viscosity itself can evolve in time and frequency, and in particular how to construct a rheological model for this systems. And so you can do that starting also usually from a kinetic description, and then you can. Now I'm noticing that the blue is not showing, but it's okay. It's just aesthetics. You can write for this normal and shear sectors. You can write a sort of a coupled Maxwell model, where now the evolution of your normal shear sector of your stress tensor and the normal difference evolve coupled, and the coupling is governed by the rotation frequency of your constituents, which also means that now we can think of controlling our systems, or we can control our material not only by the applied driving, as usually this which you think about allows response, but you can also play, at least in theory, and we will show you how we're thinking about this. You can also think of controlling the microscopy of your particles, which will then course grain on having control on this coupling at the rheological model. And at least in an abstract sense, you can think that in principle this coupling might also be not only made a function of time, that you can either actuate or have some active response from the constituents themselves, but it might, at least theoretically, it can also depend on the overall stress state of your system. So we were looking into this, and to make things a bit simpler, we are just considering that the driving is done purely on the shear sector. So we have also here introduced the Deborah number of the oscillation of your system, so the frequency at which you shear your system, and the Weissenberg number for the amplitude on which you do it. But even before looking into parametric control itself, the simple fact that now your Maxwell model is coupled, but in a sense, you can think of it as your Maxwell model is now second order, or you have more poles on your transfer functions. Well, we have one more, you have two instead of one, which already makes the viscoelastic response of material quite interesting, because now you can make your elasticity both in the shear and the normal sectors, and throughout the slides, normal differences will be represented by this more reddish color, and the shear ones to the more blue that sometimes don't see very well. But so it's already interesting by itself to control the position of the poles, that you can play a bit on the effective viscoelastic response of your material. But as I was telling you, we in particular want to go beyond this and see what happens if you evolve your coupling on time and on stress. And for instance, if you look into some parametric actuations of your system, so if you imagine that now your rotating frequency is modulated such that your particles rotate more slowly and then faster, and then slowly and faster, you can unlock further phenomenology on the system. And notice that now I'm introducing a new time scale. So at overall we will have 3 time scales in the system. So we will have the driving frequency, the external driving of your shear. You have the rotation, central rotation frequency, and you have this this modulation period on your system. And so what we found is that not only this parametric excitation is still stable in the systems under consideration, but choosing, of course, the interplay between the driving and the modulation can lead you to control the fluidization state of your system, in the sense that now your dynamic, your time evolving viscosity and elasticity change with the modulation. So what I'm showing here is the elastic moduli. Again, blue is shear sector and red is normal sector. And now during modulation period, you can go from a state of higher elasticity and lower viscosity to the opposite. And you can play choosing your modulation or designing your modulation. You can play a bit on if your system lays most of the time on the more fluidized state, or in the more fluidized in the sense of more viscous state, or in the more elastic state, just by choosing the modulation that you impose. So this is what you can do simply, or this simple thinking on parametric control or time controlling your rotation frequency. But you can also think, as I was telling you, that okay, let's suppose that somehow my constituents also feel the stress around them, and that they, for instance, they only rotate above a critical stress, which in a way corresponds to having some sort of a Bingham-like response. And when you do that, and note that here we are only imposing control on the rotation or on the odd sector. But when you look to your stress to strain response, you attain quite flat, almost quasi-plastic responses of your stress on the shear sector, just from the coupling and the nonlinear or the step response of your system. So this means that you can attain pseudoplasticity just by controlling not your shear degrees of freedom, if you wish, but just locking or unlocking the rotation, internal rotations of your constituents. And you can, of course, study this throughout throughout the excitation frequency, and also the amplitude to which you drive your system, and do a sweep of these parameters, and then choose exactly the sweet spot that you wish to have higher or lower degrees of this pseudoplasticity induced on our system. So this would be very, very interesting, in our opinion, to design the future system that would behave like this, so that you can tune and control and design exactly their viscoelastic properties. And so, until now, I've been talking about simulations, about modeling, but we want to go a bit further than that. Even though I do mostly theory, I also enjoy going downstairs to the lab sometimes. And so now I will show you a bit of how can we think of building a system like this. And if you remember in the beginning, I showed you already some movies of other groups doing similar stuff. So, as I was saying, we need, this talk, we need something that rotates. And it would be way more interesting if, instead of relying on actuated magnetic colloids or something like that, if we can rely on particles that self-rotate and that you don't need to drive externally. So in a way, we want to look into something active and hybrid. And this is exactly a video of our system So what we are using here is a suspension of anomalous linearity, which for those who don't know, and 2 years ago I had no idea as well, are these cute little rheology that usually in the wild, the ponds that we find, have 2 flagella, and which allow them to swim, to propel. And you can develop models of this very simple organism that you can understand their equations of motion if you think of them having these 2 spoke slugs on each flagellum and also a compensating one in the center. So, but this is the wild type, and as you can already tell, this is interesting on itself, but it's not chiral, so there's nothing rotating here. But thankfully for us, there's not only the wild type, but there's also a mutant strain that only have 1 flagellum, and interestingly, and through the magic of genetic editing, it always have the same flagellum. So it always has it always has the trans flagellum, which means that 1, they rotate because now they can propel, and more importantly for us, they all rotate in the same direction. And now, of course, to understand the system, or at least to model at the particle level, you can consider just 2 point forces, but then of course you need also to compensate and to think of a point torque also applied in your system. So this is what we have been studying lately, doing these suspensions, and as I was telling you, they all rotate in the same direction, or at least depending on whether they are near. So we studied this on a nanofluidic, microfluidic channel, and so then there's 2 populations: the one that sediment near the bottom and the ones that float near to the top wall, but at either population they have very well defined rotation, so they all, in a sense, their angular momentum always points towards the inside of the fluid. And also it's an interesting model, and it's an interesting experimental setup, because we are very lucky to have a quite nice separation of time scales in our system. So they beat their flagella quite fast, around 50 hertz. But then this translates to a quite slower rotation. And then, and you'll see it in a minute, they also, due to this rotation, they also start to have pair interactions, and they start to orbit each other, but also this orbiting is also way slower than the previous time scales. Which means that we can, thankfully, average out the flagellar beating and just focus in the 2 slower time scales. And so, as I was, so in this video you probably can barely see the flagellum, but it's somewhat here now. And as you can see, it beats way faster than the rotation. Then to study these suspensions, what we do, as I was telling you, is to consider that the whole organism is well modeled, and I will show you a comparison in a bit, is well modeled by a pair of point forces located at some distance L comparable to the flagellum, or to the average effective flagellar length, and also compensated by a point torque. Because, and I should mention that we are in a Stokesian limit, because so you have to, so it's torque free, it's always torque and force free in your system, so you have to balance your Green's functions. And then you can average over the rotation, and you have an effective velocity field in the radial and azimuthal component that actually decays quite fast into the decay R-4 and R-6. And that also depends on this phi angle of the force relatively to the flagellum. And with that we can model the clavinomonas, but you can also model clavinomonas that have an effective attractive radial component, but you can, because they are what you are calling the system, they are pullers. But we can also study, and there are also other organisms that are pushers, that they repel each other. Also interestingly, because of this fact that we are at a low Reynolds limit, there's this inversion of chirality on your system, in the sense that the effective rotation of your flow that neighboring cells feel is not on the same direction of the cell itself, because you need to balance this the torque by the drag of the body itself. So weirdly, or in a sense, if you would think naively, but weirdly, if they rotate in one direction, but then the dynamics is governed by the other, by the opposite chirality. But so I was telling you that we implement this model, then we can add some tracer particles and see how good our model is. And I'm showing you here our experimental average flow fields that we observe in the lab, and the results compared to our model, both just looking at the power stroke, or at least the most we can, defining where the power stroke is in the experiment, and with a full rotation average. And here is the full, not just the far field response that I showed you, but also the full model that you can see that you can retrieve quite well, I would say, the effect or the response on these systems. But having understood this single cell, now it's interesting to start looking at this in the lab, what happens with the interactions, and what happens in when the cells start to talk, in a sense, to talk to each other. And we see already so this is the same video, but here is just running faster to make more obvious that while they spin in one direction, they start orbiting the other in the opposite direction, and so also it's clear that they are interacting, and that we are hoping to we are having hope to have this in future this macroscopic effect. And you can see not only the pairwise interaction, but also bigger clusters of cells. So you can also find stable configurations of 3 cells. Here, certain like 7 cells. And in particular, I really like this top video here, where you have a quite stable configuration of these 6 cells in this triangular lattice, which is also interesting that because of the balance of the interactions, the smaller cells are placed on the vertices. And so we, throughout the experiment, we see always this overall clockwise orbiting rotation. And an important point here is that because they still have this interacting component, this inwards radial component, we were a bit worried in the beginning if we would end up with a crystal, like the work with the sea urchin embryos. But actually, because we believe both of the noise on the beating of the flagellum, but also the fact that this is a way more polydispersed system compared to the sea urchins, we don't. We end up having a gas or liquid of our cells, because even though you have these somewhat stable clusters, then you don't grow much further than that, and you can start to look. And that's what we are doing actually right now, to start to look into the denser suspensions of these cells. And we haven't yet measured for viscosity. We have already some hints of odd transport coefficients, in the sense that we have a hint already of odd diffusivity, in the sense that if you measure the transverse current, or the transverse flux of cells over a density gradient, you see that the transverse current is way higher than the direct parallel current on our system. So this gives us hope that in the next steps we will be able to measure also hot viscosity and this overall behavior that I talked and that I showed you in the beginning of my talk. And so this starts to bring me to the end. And so just to wrap up the three main sections that we saw. So I talked to you about this hot viscosity and how we can already see interesting or strange effects on surface waves. In particular, this drift and this direct parallel motion that you can think in the future of controlling. Then, even more interesting in a way, to also couple this with the rheological model that I showed you, where you also take into account the time response of the viscosity itself. And lastly, we showed you briefly what we have in the lab right now, that we truly believe that in some months at least, let's see how lab goes, we will have also a biosuspension that behaves exactly as I was describing in the theoretical part of the talk. Some future direction that we can take from here is also to think, and we have other people working on that not only thinking about algae or not only thinking about biological systems, but we can also think about fully synthetic materials. And what I mean by that is built, for instance, from small robots. And it would also be quite interesting to study, and we are working on that right now, is to mix not only the glamines that rotate in one direction, but also mix them with either some passive particles or mix them also with counter-rotating particles. We have already in the lab other organisms that rotate in the other direction. It's just a bit tricky to make them be happily in the same fluid, in the same suspension. And lastly, so far I've focused in viscosity, which implies that I'm so far only looking into momentum transport, or linear momentum transport. But it would be critical also to look into the transport of angular momentum in the system, assuming that not all of your particles rotate at the same rate. And you allow for non-uniformity on the rotation itself. And with that, I would like also to thank and to mention the team, Mazi, Isabel, and Ubu on the theory and simulation, and Holly, Anna, and Titouan on making all this possible in the lab. Thank you very much.
Great talk. So now we're open for questions.
Considering your final example, the experimental setup. Why do two of these bacteria, they come together? What is the attractive mechanism? Because if they are rotating in the same direction for purely hydrodynamic arguments, I would say that they move away, right? Because they have the same sense of rotation, and therefore you have an effective difference in velocity at the interface between the vortices that they form. Right? If they were moving and
if they were just vortices,
then I would expect them to come together. Here, and on top of that, they have that flagella that in principle eventually will collide and move them apart.
Yes.
So
Yes.
what is the mechanism?
So there are two parts. So of course starting with the last, of course the flagella then impose that there is some steric propulsion, and which in a sense, if you look, is, well, you can have a video here where you can see the flagella, I think. Yeah, here you can see the flagellum more clearly, hopefully. And yeah, so the flagellum set the minimal distance that they can. That for sure. Or even here. But on the attraction part, the trick is that they are not pure vortices. And maybe I went too quickly over that. But in our model, and also then it seems to agree quite well. So there's also this radial component of your Stokes, right? Because the force that the flagellum exerts is not exactly perpendicular to the flagellum. So there's this radial component on top of the vortex interaction. And so the radial interaction comes exactly from the projection of this force. Because of course, when you average over this rotation, you still have a bit of that projection.
Okay, so it's not a pure vortex, but
No.
then what keeps them together? Because they are orbiting, so there is some kind of attraction between the two, right?
No, yeah, the attraction comes from here, and maybe I should have drawn this the other way. So because in the clown this phi is larger than pi over 2.
Mm-hmm.
So there's this attraction from the fact that the Stokeslets are not aligned with the flagellum,
Mm-hmm.
and so they attract, and then they, at a certain point, at roughly 2L, they start seeing each other flagellum, and then they stop. Yeah.
Thank you. I was thinking about if you want to describe this macroscopic collection of those objects, let's call a gas of this.
Mm-hmm.
So they would, in principle, you could start with your say microscopic Remontovich approach or whatever,
Right.
and then you couple to the fluid because then it's suspended this biofield. So the mean field force between these particles is provided by the fluid somehow, right? So you self-consistently
Self-consistent, but you need to think, and it's a bit of a shift that I also had to do. Everything here is, at least in this system, everything here is quite overlap.
yes,
So-
this is
And
fluid itself. Will
yes, but the at least our approach is, well, it's overlap and it's linear because you're in Stokes limit. So what we are doing is you just superimposing naively, just superimposing your velocity fields of each. So what I mean is that you don't simulate or you don't evolve. You could if you have a lot of power, of computing power, right? But you don't. What I mean is that you don't solve the flow and the advection of the particles.
I agree, but this is somehow the propagator of.
Yeah, exactly.
So
Yeah.
you put this propagator and your gauge boson for your interaction somehow.
Yeah.
You have the language that you understand.
Yeah.
So this particle is field here then is also dynamic because it's coupled to some Navier-Stokes equation. You understand?
Yes,
So
but
this,
it's solved. Not that's the thing. Not Navier-Stokes. And
so this is the debatic elimination result.
in a sense,
Okay.
yes. So the structure. So you get to this you when you use already the Green's functions of your Stokes problem.
Okay.
And so in a sense, what I'm representing here is that you imagine that you have these 2 point forces and this torque. Then you look. I don't think I have an extra slide for that, but I can show you also later. Then you work out they're already known the Green's functions of both the point forces and torques. And that's how you construct first this, and then you average it out over a rotation. So the solution of the Navier-Stokes is already ingrained on the propagators, on the Green's function that you are using to get here.
But this is valid for one particle?
Yes.
So you have two particles and then you want to do mid field or whatever. The thing is this potential will be changed and will be a function of the distance between these particles. Let's say the density.
Yes. Yes.
And that's why we couple to the kinetic equation, because density now is the integral of your phase space distribution function, which evolves according to some Boltzmann equation.
Yeah.
In that sense, that's why I'm trying to let's say, formally obtain, because in real life, I suspect, as you mentioned, these particles don't have all the same velocity. So you have to integrate out over the distribution of angular velocities.
Yeah.
And this will be where the reality comes from.
Of course, they won't have all the same, but it will all be of this.
Of course,
form.
I totally agree.
Yeah.
Totally. But then it will be pondered, averaged over the
Exactly,
distributions.
Which then is trickier on
Objectivity.
And then it's also trickier if you also want to start to include all dispersion.
Okay.
But yeah.
But that's very, very beautiful for you.
Questions? Maybe I'll.
No, it's a simple question. In the same spirit, can you come up with an effective interaction potential between the particles with angular dependence that you then could add into a simple Brownian motion dynamics and try to come up with
we are looking into that.
Okay.
Especially we can.
Because it would be a simpler way to try to justify the critical or the characteristic size of these clusters, right?
Yes, exactly. Yeah, but I agree with you that for that, and we are starting to do that. For that, you need to go before. You shouldn't already average. You should have the polar dynamic. Yes, and we are looking into that. It seems quite interesting. We are. Also, it's interesting in several aspects, I would say. We seem to, more or less as expected, to get to a generalized XY, in the sense that we have both polar and nematic ordering. And so far we are looking, to start with something, into the synchronization of that. Because we already have some
Yeah, this gives you the coupling between the rotational degree.
yeah, exactly. Yeah.
Can I ask a quick one, just to clarify on the first part, because I missed it. What sets the side they're moving? So because you said after that initially they go to the left, after some critical point they go to the right.
You mean
the surface wave.
Yes.
So what sets the left-right symmetry, or
what we see, and we have more to discuss on that, but what we see is that above, I think it's 0.3, so above a critical ratio of surface tension and of viscosity, you switch.
yeah, but okay, but is it always first left and then right, or do you
No, not
just
first. I mean, it's on the parameter of your system. So if your system is with low odd viscosity, in a sense it follows the wave. And if you increase your odd viscosity,
It goes against
the wave.
And then just a curiosity on the last part, because you said it was different from the sea urchin system where they crystallize they put big clusters. So, what is the difference there? What is the biggest difference between the two systems? Or I don't know if we discussed that.
Going bio. If I understand correctly, the theoretical work, I would say that there are three important differences. First, the attractive component is very much, if I remember correctly, is very much also induced by the free surface. They are near a free surface, so we have a lot of capillary effect on that. Second, they are not flagellated, they are ciliated, and in the sense that the important, I would say, thing about the clam is that the length scale of your flagellum and your body is roughly the same. And in the sea urchins, the body is way bigger than the cilium. So there's not this you cannot see this interesting interplay between the cilium. And lastly, because, well, in a sense because nature, the way you grow your sea urchins we are talking sea urchins embryos. They are all pollinated at the same time. They are way more consistent on sizes. They are, of course, they are way more monodispersed than our algae, which I think plays a significant role on the dispersion.
Just a final comment before you go, because I cannot resist. This transition here, if you think about the surface as edge state, somehow, okay, in some manifold that is crazy, does it signal that you are facing some sort of topological phase transition?
Maybe. It's not very clear to me how you would frame it.
This is law of the spirit.
No, in Suslov spirit, no. In Suslov spirit, no. Or at least I don't. Answering quickly, you don't have any gap to cross here. At least not in the waves. And then also the vertical edge state is purely decaying, right? Or what I mean by this is that it seems complicated to me to think of a framework in that direction.
Great. There are no more questions. Let's thank the speaker again. And if anyone wants to chat for discussions, we're going to the cantina next to discuss stuff there.